can summarize the chromosomal theory of inheritance in a very simple way like this. The behavior of chromosomes during meiosis 1 is parallel to the behavior of genes. This is the gist of the chromosomal theory of inheritance. Let us apply this idea to explain the monohybrid inheritance. In monohybrid inheritance, let us go back to the same example of the pea plants, the cross between tall and dwarf pea plants. In the F1 generation, all the resulting plants were tall. When these tall plants were allowed to undergo self-pollination in the F2 generation, tall plants and dwarf plants resulted. In the phenotypic ratio of 3:1, and genotypic ratio of 1:2:1. So he represented the tall plant as capital T, dwarf plant as small t. Only one factor, according to Mendel, is passed on to the gamete. So the gamete will be capital T here, small t here. The combination of these two will be capital T, small t, capital T dominant, small t recessive, so all tall plants. And in the next generation, selfing, it is here that he talked about segregation. The two factors are staying together in the F1 generation but segregate during the formation of gametes. Factors staying together segregate during formation of gametes. So two types of gametes. 4 combinations are possible. Three tall plants and one dwarf plant, one homozygous tall plant, two heterozygous tall plants, and one homozygous dwarf plant. Suppose we represent this idea based on the chromosomal theory of inheritance. let us say this is one pair of chromosomes, one pair of homologous chromosomes in the tall plant. These are the two chromatids, this is the centromere, and let us say the gene responsible for length of the stem, is located on the same locus in the two chromosomes. This is a homozygous tall plant. Therefore, on this locus you have a capital T here, capital T here, into a dwarf plant. In the dwarf plant also, the same chromosome in the same locus carries the alleles responsible for length of the stem. This is a homozygous recessive tall plant. So, in these loci it should have small t, small t. What will be the gamete now? Of these two homologous chromosomes, there is separation of these homologous chromosomes during meiosis. So, you will see the two homologous chromosomes separating Similarly here, the two homologous chromosomes separating so these are the gametes. In other words, 50% of the gametes carrying capital T, 50% of the gametes carrying small t. It can be this with this, or this with this, this with this, or this with this. Any combination, all that you are going to get will be in the resulting gamete, the resulting zygote. You will have in the same locus one homologous chromosome carrying the dominant gene, another homologous chromosome carrying the recessive gene. This plant is going to be a tall plant because this gene is dominant over this allele. Now, when there is selfing, suppose I write this combination twice. Heterozygous tall plant. Now, during the formation of gametes, see what is going to happen. One homologous chromosome goes to 50% of the gametes, the other homologous chromosome goes to another 50% of the gametes. That means the homologous chromosomes are segregating, therefore the genes are segregating, the factors are segregating. So you will have a gamete two types of gametes, capital T, small t. Similarly here, two types of gametes, capital T, small t. So 2 into 2, these 4 combinations you will get. When this gamete fuses with this gamete, it will be capital T, capital T. When this gamete fuses with this gamete, it will be capital T, small t. When this gamete fuses with this gamete, it will be capital T, small t. And when this gamete fuses with this gamete, it will be small t, small t. This is homozygous tall plant, heterozygous tall plants, homozygous dwarf plant. So it is the chromosomes that are segregating, therefore the genes present on them are also segregating. This is how chromosomal theory explains the idea of segregation.